In this video, we're going to learn about how the most common neurotransmitter in the body functions to regulate the neurons that it regulates. This is a chemical called acetylcholine, the name for which will become important because it describes the two halves of the molecule that we see here. This is what we call the acetyl half because its structure is based on acetic acid, CH3COOH, and this is the second half, the choline half. So that's the origin of the name, which will be important later when we talk about how it's regulated. Now, as the most common neurotransmitter in the body, it has the exceptionally important role of regulating the motor neurons that control the skeletal muscles in the body. This is particularly important because skeletal muscles are used in voluntary motion, which means every time you try to move your hands, feet, arms, or legs, you are using acetylcholine to mediate that motion. Now, acetylcholine is probably the best example of what we call an excitatory neurotransmitter, meaning that unlike inhibitory neurotransmitters, it activates rather than inhibits the movement of skeletal muscles that it regulates. Now, synapses that are regulated by acetylcholine are what we call cholinergic synapses, in obvious reference to the name of the neurotransmitter that regulates them. Now, if we take a look at the actual axon terminus of a cholinergic synapse, we can see that all of the components that we would expect to find are present. We can see synaptic vesicles that are carrying acetylcholine, which, like most neurotransmitters, is assembled in the cell body and transported down the axon to the axon terminus, but via the cell's cytoskeletal transportation network. Now, the regulation within the axon terminus requires a little bit more specificity, specifically regulating the exocytosis of the synaptic vesicle that occurs when action potential makes it down to the axon terminus. So how exactly does action potential regulate this process of exocytosis? Well, just as a little bit of background, remember that action potential is caused when enough sodium ions diffuse inside of the neuron's cytoplasm and therefore cause both the inside and the outside of the neuron's membrane to become positively charged, and this causes cell membranes, specifically voltage-gated sodium channel proteins, to adopt an open conformation and allow even more sodium to come into the neuron. All of this should be old news as of the action potential videos. Now, this is important to talk about because the same mechanism also affects channel proteins that transport calcium ions that are only found at axon termini. And just like how high sodium concentration causes voltage-gated sodium channels to open, these also cause calcium channels to open specifically at the axon terminus, meaning that calcium is able to enter the axon terminus. Now, calcium is not directly involved in the process of action potential, but is very, very critical in regulating exocytosis. So on these synaptic vesicles that contain acetylcholine, there are specific receptor proteins that match receptor proteins that are found on the inside of the axon's membrane, and when these receptor proteins bind, it triggers the process of exocytosis. The problem is, as we can see from the haphazard diagram here, the structure and shape is not complementary enough in order for these receptor proteins to bind to each other. But what they can do is bind the calcium that is now diffused into the inside of the axon terminus. And when calcium binds to this receptor protein, it uses its charge to cause a conformational change in this membrane protein in order to make it more complementary to the protein that triggers exocytosis. So it is this conformational change on the proteins that are found on synaptic vesicles that allows exocytosis in order to occur better. And when this process is able to happen, it causes the acetylcholine that is contained within these vesicles to be released into the synaptic cleft. This is how action potential mediates this entire process, because without the depolarization of the membrane, these calcium channels would stay closed, and synaptic vesicles have no way of binding to their appropriate receptors without this key conformational change that mediates the process. Now, when acetylcholine is released into the synaptic cleft, it is able to bind to receptor proteins that are found on the postsynaptic neurons' dendrites, and we know that acetylcholine is an excitatory neurotransmitter, which means that when these receptor proteins are bound, they trigger a conformational change that causes Na plus channel proteins that are found on the postsynaptic dendrites to open, therefore allowing Na plus to flow into the dendrites of the postsynaptic neurons. Now, when this happens, the entire beginning of action potential begins once again. This increase in Na plus is what is necessary to cause the voltage increase from resting voltage at negative 70 up to our threshold voltage of negative 55, and this sodium that enters is enough to cause the conformational changes in the voltage-gated Na plus channel proteins that are found on the postsynaptic dendrite to open, allowing even more Na plus to flow from the outside in the synaptic cleft inside to the dendrites, thereby causing action potential to begin once again. Now, this is the most important part of acetylcholinergic neurons being regulated, the idea that we do not want acetylcholine to stay here because we do not want this neuron here that directly regulates muscles in order to be constantly active, because then your muscles would contract without being able to relax and you would be going through a muscle spasm. So the way that acetylcholine is regulated is actually fairly unique compared to all other neurotransmitters, in that it is pretty much the only neurotransmitter that uses a digestive enzyme in order to break its structure. And this digestive enzyme, whose name will take a little bit of time to write out here, actually tells us exactly what the enzyme does. And that enzyme is called acetylcholinesterase, one word, which we can abbreviate as ACHE for acetylcholinesterase. And this is an enzyme that catalyzes a hydrolysis reaction, meaning that it hydrolyzes acetylcholine, specifically the ester linkage between the acetyl half and the choline half. And after it grabs a water molecule, breaks this bond, and adds hydrogen to the to the oxygen here, and OH to the choline half here, we would end up with the acetate half, the base form of acetic acid, and the choline half, which we can see here. Now, after acetylcholine is broken, obviously the two separate molecules will not be able to fit in the active site anymore, which means if the active site of acetylcholine's receptor is not bound, that means that the neurotransmitter-gated sodium channel is not going to stay open either, and as a result, it closes, and Na plus is no longer able to get into the postsynaptic dendrites, and our action potential process has just been stopped. Now, the elegance of this particular situation is that even though acetyl or acetate and choline, the two halves that make up acetylcholine, are broken, that doesn't mean that they are wasted, because acetyl and choline can actually be recycled. And the way that we do that is that acetyl and choline, or acetate and choline rather, are reabsorbed into the presynaptic neuron, so into the presynaptic axon terminus, and once they are brought back in to the presynaptic axon terminus, they can be transported back to where a dehydration synthesis enzyme is located in order for it to be reassembled and ultimately recycled, which allows this process to effectively continue indefinitely. This concludes our video on acetylcholine. In the next video, which will be shorter, we're going to take a look at a different hormone's method of regulation, which is a little bit simpler, and this is a hormone that regulates involuntary processes rather than those of voluntary ones.